Hello everybody. Today I am going to discuss about the diagnostic part of the hemolytic anemia, and this is the sequel 4 of this hemolytic anemia series. If you are new to my channel, don't forget to subscribe my channel, like and share my videos, and subscribe to get the notification of the daily uploaded new educational videos. So, how the diagnosis of hemolytic anemia is done. The diagnosis of hemolytic anemia, which is in brief HA, may be suspected on the basis of group of symptoms. So, only one symptom cannot diagnose the hemolytic anemia. So, there should be some group of symptoms which will notify the presence of the hemolytic anemia in a particular person. So, what are those symptoms? It is predominantly based on the occurrence of anemia. First of all, a person should have anemia or the symptoms of the anemia, then we will classify whether it belongs to hemolytic anemia or not. An increased proportion of immature red cells, that is reticulocytes. Hemolytic anemia means absence or very few amount of red cells are present there along with the very few amount of hemoglobin. So if there is no mature red blood cells, what will be present in the circulation? Immature red blood cells, that means the reticulocytes, and a decreased level of haptoglobin. What is this haptoglobin? It is a protein that binds the free hemoglobin. So there are three symptoms which can be grouped to diagnose whether it's a hemolytic anemia or not. Number one, occurrence of anemia. Number two, an increased proportion of immature means reticulocytes. Number three, decreased level of haptoglobin. Now, what are the basic diagnostic testing? Number one is bilirubin level. So testing the level of bilirubin in blood actually measures the level of red blood cell hemoglobin that liver has broken down and processed. Means how the liver functions. Here comes the liver function also. Bilirubin level is the result, liver function is the cause. This test measures the level of protein, liver enzymes and the bilirubin in the blood. So next, hemoglobin level. Testing of the hemoglobin level indirectly reflects the amount of circulating red blood cells in a person. Naturally, if we know that how much amount of hemoglobin is present in RBC, then how much amount of RBC is present in the blood, then generally we can guess the amount of the hemoglobin level in the blood. It is done by measuring the oxygen carrying protein within the RBC. So we all know that hemoglobin is the pigment which carries the oxygen all throughout our body. Now urine test. This test is done to look for the presence of RBC breakdown. So whether the broken RBC came to the urine or not. So here the four tests are very important. So bilirubin level testing, hemoglobin level testing, liver function and the urine test. So what are the other tests? Next is reticulocyte count, which is very important. This test measures how many immature RBC the body is producing. The anemia of hemolysis usually is normocytic. Normocytic means the cell size is normal. Normo means normal. Cytic cell size. Although a marked reticulocytosis means increased in the reticulocyte count, can lead to an increased measurement of MCV, means mean corpuscular volume, because the average mean corpuscular volume of reticulocyte is 150 femtoliter. So reticulocyte count, whether there is an occurrence of reticulocytosis, it is a very important diagnostic feature of the hemolytic anemia. Next, review of peripheral blood smear. Direct examination of blood drop under a microscope as a smear may demonstrate red blood cell fragments called cystocytes. So there may be some red blood cell fragments like cystocytes. Red blood cells look like spheres called the spherocytes and or RBC missing small pieces like bite cells. So, cystocytes, spherocytes and bite cells. These three can be found in the peripheral blood smear if reviewed for the diagnosis of the hemolytic anemia. Examination of the white blood cells and the platelets for coexisting hematogenic or malignant disorder is also essential. Means whether there is only hemolytic anemia is present in the person or some other problems are also there. Next, biopsy. In some cases, doctor may ask for a bone marrow aspiration or biopsy. This test can provide information about how many RBCs are being made or their shape. So a detail of the RBC can be found from this biopsy report. Other tests includes like test for bilirubin and lactate dehydrogenase. This is very important, in which both are found to be increased. Then test for haptoglobin, in which the level decreases. Actually, what happens? Lactate dehydrogenase and hemoglobin, these two are released into the circulation when red blood cells are destroyed. Okay. Now this liberated hemoglobin, it is converted into unconjugated bilirubin in the spleen or may be bound in the plasma by haptoglobin. Now this hemoglobin haptoglobin complex is cleared quickly by the liver, leading to a low or undetectable haptoglobin levels. So this is the underlying physiology. Apart from these two, direct Coombs test to evaluate antibody binding of to red blood cells suggesting AHA, autoimmune hemolytic anemia. So there are references for your further study. Thank you very much for your patient listening. This sequel will continue to part 5. So long. Bye bye.